Welcome back to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from neuroscientists across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, and for more info on the talks covered in this seminar series and all things neuro-related here at Cambridge, please follow us on Twitter at CamNeuro and follow the links below. Welcome everybody to what is the last in the current series of the Cambridge Neuroscience Interdisciplinary Seminar Series. As many of you can testify, we've had a really excellent series this year, and we're delighted to end it with Dr. Caroline Williams-Gray. Caroline is a Principal Research Associate at the Department of Clinical Neurosciences here at Cambridge. She's also an honorary consultant neurologist specializing in Parkinson's disease and movement disorders. She leads a translational research laboratory which specializes in the investigation of the clinical and biological heterogeneity in Parkinson's disease. So today she's going to tell us about her current work. And over to you, Caroline, and thank you so much.
Thank you very much for the kind introduction, and I'm very, very happy to be here today to give the closing talk in the program for the year. So I'm going to talk to you about immunosuppression in Parkinson's disease, which might sound like a slightly crazy idea for a new treatment, but hopefully by the end of 40 minutes or so, you'll agree with me that it's not such a silly thing to consider. So I just wanted to start by highlighting the therapeutic challenge that we have in Parkinson's disease. So this is a condition which, as many of you will know, is characterized pathologically in terms of a loss of dopamine-producing cells in the substantia nigra. So we're looking here at a post-mortem section through the midbrain in a control. Let me just see if I can get a pointer, bear with me. So this is a control midbrain. You can see here these pigmented cells of the substantia nigra, which are the dopamine-producing cells. And here in the midbrain of a Parkinson's brain, the substantia nigra is pale because we've lost those pigmented cells. And this dopaminergic neurodegeneration in the nigrostriatal system leads to these typical movement symptoms of slowness and stiffness and tremor that we see in our patients. Now, this can be ameliorated to some extent with dopamine replacing therapies. So if we restore dopamine in this nigrostriatal system, we do see some clinical improvement. So this is a treatable condition for many people, at least for the first few years. But the problem is that over time, what happens in Parkinson's disease is that a considerable proportion of patients will develop quite disabling symptoms, postural instability, falls, and in many cases, a dementia. So these problems affect about 3/4 of our patients by 10 years into the condition. And these problems are due to neurodegeneration outside of that nigrostriatal dopaminergic system. So this is due to widespread neurodegeneration affecting cortical regions of the brain. And the dopamine replacing therapies don't help these sort of symptoms because they're not dopamine, they're not due to this dopaminergic loss in the nigra. So the question is then, how can we help these symptoms? How can we slow progression to these disabling problems that have such a major impact on quality of life? And there are various different therapeutic strategies that people are considering, as you may know, targeting things like aggregation of proteins in the brain, targeting lysosomal function, targeting mitochondrial function. And another big area of interest in recent years has been targeting the immune system, which is what I'm going to focus on today. And for me as a clinician, this is a particularly exciting target because potentially it is highly tractable therapeutically. We already have very effective immunosuppressive therapies that we can potentially reposition for use in Parkinson's disease. So why do we think that targeting immune activation in Parkinson's is a good idea? Well, evidence has been accumulating for the past 20 or 30 years that the immune system is activated in PD. So it was back in the 1980s that McGeer and colleagues first published data in looking at post-mortem brains showing that there was microglial activation in the substantia nigra, and numerous post-mortem studies since then have similarly found evidence of neuroinflammation in the post-mortem brain. In vivo, we can use PET ligands, so we can use TSPO ligands that bind to activated microglia in the brain. And these sorts of studies have demonstrated, again, that there is neuroinflammation happening even in the earliest stages of Parkinson's disease. There's also emerging evidence that the peripheral immune system is activated in Parkinson's, and I'm going to come on to tell you a bit more about that later in the talk. But these things clearly happen: inflammation in the brain, inflammation in the periphery. The question is, how are these things linked? So this relationship between peripheral immune activation and central neuropathology is unclear. It's now, however, widely accepted that the brain isn't the immunologically privileged site that we thought it was. So we do know that peripheral immune cells can infiltrate into the brain. We know that peripheral cytokines can influence brain function. This becomes clear when people have peripheral infections. If you have a viral infection affecting you systemically, then you experience symptoms such as fatigue and nausea and fever, which are brain mediated, which is centrally mediated, providing evidence that actually what happens in your peripheral immune system does influence what happens in your brain. So on the basis of that, the hypothesis is that not only local brain inflammation is going to be important in Parkinson's, but also what's happening in terms of the systemic immune response. So we think that the systemic immune response might be driving both early disease onset and faster disease progression in Parkinson's disease. So over the next half an hour or so, I'm going to talk through some of the work from my lab that we've been doing over the past few years, investigating this hypothesis. And as I said at the beginning, trying to convince you that that we have a good rationale for clinical trials targeting the immune system. So I'm going to cover the next 4 questions over the next half an hour or so. I'm going to talk about whether immune factors are associated with Parkinson's disease risk. I'm going to consider whether immune activation is linked to the type of Parkinson's patients have and the way their Parkinson's progresses. I'm going to then think a bit more about which particular immune cell subsets are involved, and then finally talk about how we're translating those findings to clinical trials. So firstly, this question of whether immune factors are associated with risk of developing Parkinson's disease. So the strongest evidence for this really comes from genetic studies. And we started looking at this back in 2009. We did a collaborative study with our colleagues who are interested in the genetics of MS. So we were doing this work with Steve Saucer's lab here in Cambridge. And they were very interested in HLA-DR and the fact that common variation in HLA-DR is associated with risk of multiple sclerosis. And we thought we'd have a look at the same variants that were associated with MS and see if there were any links to Parkinson's disease. So we looked at a cohort of just over 500 of our Parkinson's patients and a much larger group, about 3,000 controls. And what we found was that there did seem to be this association between a common genetic variant in HLA-DR and your likelihood of having Parkinson's disease. Now, HLA-DR encodes MHC class II proteins. These are critical proteins in the adaptive immune system. They're involved in presentation of antigen to peripheral immune cells. So it seems that this predetermined variation in the function of the adaptive immune system has this influence on increasing risk of getting Parkinson's disease. And that finding has since been replicated in numerous genome-wide association studies over the past few years. We also know that numerous other immune genes, both in the adaptive and the innate system, seem to be related to PD risk, and there's too much literature to go into in any detail. The other bit of evidence comes from these sorts of pathway analyses, which look at common risk variants associated with PD that have been picked up in large-scale genome-wide association studies, and tries to map these common genetic variants onto particular biological pathways. And those sorts of studies have consistently implicated innate and adaptive immune pathways. And the other area of the other type of study, I should say, that really sort of confirms this idea that the immune system is linked to your risk of getting Parkinson's disease are epidemiological studies. So there have been a few epidemiological studies now looking at links between inflammatory conditions and risk of Parkinson's disease, and they suggest there does seem to be a positive association. And studies have also looked at the association between immunosuppressant drug use and risk of subsequent PD. So I just wanted to flag up this particular study, which was a study done in the US about 5 years ago. So they looked at about 50,000 people with incidence or newly diagnosed Parkinson's disease and a similar number of controls. And this is a case control study when they then looked back at these groups and looked at how many of the individuals in these different groups had used immunosuppressive drugs for more than a few months. And what they found was in the patients with Parkinson's disease, they were significantly less likely to have taken immunosuppressant drugs. And this was particularly the case for this class called inosine monophosphate dehydrogenase inhibitors. These are drugs like azathioprine, mycophenolate mofetil, which predominantly affect the peripheral immune system. So suppression of peripheral immune activation seemed to be associated with an increased risk of getting Parkinson's disease in the future. We've done some similar analysis, but using a slightly different methodology. This is as yet unpublished. This was done by an MPhil student recently. And they took data from CPRD, Clinical Practice Research Data Link, which is a big database of primary care data that's collected in the UK. And from that database, we extracted data on patients who had taken immunosuppressive drugs and then looked at their subsequent incidence of Parkinson's disease over the next 20 years. So comparing 64,000 people who've received immunosuppression with the population, the rest of the population on that database of 8.4 million non-immunosuppressed individuals, we found that there did seem to be this significantly lower risk of getting Parkinson's disease in the immunosuppressed individuals. And again, the risk reduction was pretty similar to what they've seen in this previous study. So about a third less likely to get PD if you've received immunosuppressive drugs for any length of time. Okay, so genetic and epidemiological data then both indicate that immune factors are linked to your risk of getting PD. But that's all very well, but when we come to think about treating patients, what's perhaps more relevant to is whether immune activation has an impact once you have PD. So that's what I'm going to just think a little bit more about now. So we've been investigating that question in a number of different ways in our PD clinic population. So we've been studying immune markers in the blood in our participants. We've been collecting cerebrospinal fluid to look at immune markers. We've been doing neuroimaging using a PET with these TSPO ligands that I mentioned earlier, which bind to activated microglia. And we've also been looking at post-mortem brain samples that have been donated by participants who've been through our PD research clinic. Okay, so one fairly straightforward way to investigate inflammation in Parkinson's disease is by measuring levels of inflammatory cytokines in the blood to see whether they differ from healthy controls and whether they're linked to disease severity or progression. Now, this is data from a study called Icicle PD, which runs as a twin centre study running here and also up in Newcastle. And we recruited 230 cases with newly diagnosed Parkinson's disease to this study about 10 years ago, as well as just over 90 healthy age-matched controls. This is cytokine data from their baseline blood samples, and we did a panel of cytokines here, pro-inflammatory cytokines. And on average, we found that inflammatory cytokine levels are a little bit higher in Parkinson's cases versus controls, but the differences here are fairly modest, but they reach significance for a few of the cytokines, IL-1 beta, IL-2, for example, TNF-alpha. What was more interesting in this data set, though, was looking at how these changes in inflammatory cytokines were linked to what happened to patients clinically over time. So we did a principal component analysis, reducing the data to pro and anti-inflammatory components. And then we stratified patients according to whether they had a pro or an anti-inflammatory cytokine profile. And what we found was that those individuals who had a more pro-inflammatory profile at the time of diagnosis had faster motor progression and more impaired cognitive function over the next 3 years of follow-up. So that's shown in these graphs here. So the red line represents individuals who've got a pro-inflammatory cytokine profile, the blue line anti-inflammatory, and this is their motor score. So higher motor score is more impaired, and you can see in that pro-inflammatory group, the motor scores are consistently going up over time, but the anti-inflammatory group seem to be relatively protected. And here these are MMSE scores, so mini mental state examination, lower score being worse, and the pro-inflammatory group have consistently worse cognitive scores over time. So what about inflammation in the brain? So a number of our patients who have been involved in our long-term cohort studies have donated their brains for research, as I mentioned earlier. And this is a study done by Nina Cooley, who was until recently a postdoc in my lab. So she's been looking at neuroinflammatory change across a number of different brain regions. She's been measuring activated microglia, looking at astrogliosis, looking at infiltration of peripheral lymphocytes and expression of various inflammatory markers, and correlating this with protein pathology in the brain, so alpha-synuclein tau and beta amyloid accumulation. And she's done this in 11 cases who have Parkinson's disease with dementia. 17 patients who have PD at a similar disease duration but without dementia, and a group of 14 controls. So the idea here is not just is there inflammation there in Parkinson's disease, but is it linked to the type of Parkinson's disease? Does it correlate with this more malignant phenotype with dementia? So here I'm showing you some images of tissue that's been stained for HLA-DR. We talked about HLA-DR in terms of genetic studies. So this is, in other words, MHC class 2, this antigen presenting molecule, which seems to be increasing expression on activated microglia. So she stained with HLA-DR and then quantified these activated microglia on the basis of their size and morphology. Here in the substantia nigra sections, they're a little bit difficult to interpret these, because in the nigra you've got neuromelanin, which stains black, so you can see the black melanin here, but also you can see some microglial staining, so the browner staining is the HLA-DR staining. And in the Parkinson's disease brain, actually we couldn't see a great deal of difference. These are Parkinson's without dementia, and they're pretty similar to controls. In the Parkinson's cases with dementia, there does seem to be a little bit more inflammation, but not reaching significance. So it seems like in the nigra, the inflammation seems sort of fairly burnt out in terms of what the microglia are doing. In the amygdala, though, there was a clearer evidence that actually there is more inflammation in the Parkinson's brain, and in particular that seems to be the case in the individuals with Parkinson's disease dementia. We also could see a correlation in the amygdala between the number of activated microglia and the amount of alpha-synuclein pathology. And this is seems quite relevant because of the fact we know the amygdala is a brain region that's involved in emotion and cognition. We saw similar findings as well in the hippocampus that I haven't included here on the slide. So it seems then that inflammation is there in the PD brain. It's more apparent in individuals who have a more malignant phenotype with dementia, and it seems to be region, it varies according to region, and is there in regions that are implicated in cognition. So Nina was also looking at expression levels of key inflammation-related proteins, and we've been particularly interested in toll-like receptors, which are thought to be critical in regulating the inflammatory response of innate immune cells, including brain microglia. So these receptors recognize either damage-associated or pathogen-associated molecular patterns. And TLR2 and TLR4 in particular have been shown, at least in vitro, to mediate the inflammatory response of microglia, both to alpha-synuclein and also to bacterial toxins. And downstream of TLR activation is activation of NF-kappa beta and the inflammasome and production of pro-inflammatory cytokines, including IL-1 beta. So she looked at expression of TLR2, TLR4, IL-1 beta in numerous brain regions, and we can see TLR4 activation seems to be increased in the Parkinson's brains in multiple regions. We also see increased expression of IL-1 beta downstream of this toll-like receptor pathway. So neuroinflammation doesn't just involve the activation of the glial cells in the brain. We also find evidence of infiltration of peripheral immune cells. So this is control and then PD putaminal tissue, which is stained for CD163, a marker of peripheral monocytes, which isn't expressed to any great extent on microglia. So here in the control brain, you can see a little bit of CD163 staining, but only in around the blood vessels. You can just see there. But here in the Parkinson's tissue, you can see not only these CD163 positive cells in the vessels, but also infiltrating into the brain parenchyma here. And then we also see evidence of peripheral T cells in the brain. And that's the case of both CD4 and CD8 positive lymphocytes. This is tissue from the substantia nigra again. So as I mentioned before, it's a little bit tricky to look at because you see the neuromelanin, which is dark staining here. So to help you out, we've just drawn little boxes around the T cells, and they're amplified here. So these here are T cells infiltrating into the brain parenchyma in Parkinson's. And the figures here show the percentage of cases that have a very sparse T cell infiltrate, so less than 10 cells, versus the proportion of cases that have a more extensive T cell infiltrate. And you can see clearly that in the Parkinson's cases, which are blue, and then the Parkinson's dementia cases, which are purple, there is this increase in T cell infiltration. And we also found that the numbers of T cells infiltrating into these regions, both the nigra and the amygdala again, were linked to the amount of alpha-synuclein pathology and also to the amount of microglial activation. Okay, so a major limitation of this sort of post-mortem work is the fact that it looks at end-stage disease. And actually what we really want to know is whether neuroinflammation and immune activation are relevant earlier on in the disease process. So we've been trying to explore this with a longitudinal study in newly diagnosed patients. And this is a study where we have used PET imaging using this ligand I've already mentioned, PK11195, binding to activation microglia in the same patients that are having the scanning. We are collecting cerebrospinal fluid to look at immune markers and also collecting blood samples, again, to look at various different immune and inflammatory markers. And the strategy with this study has been to collect these two specific groups of individuals who are either at low dementia risk or at high dementia risk. So they're all newly diagnosed, they've all been diagnosed within a year or so, but based on their tau genotype and based on their performance on a couple of cognitive tests, which we have shown to be particularly predictive of subsequent dementia risk, we can stratify them into these two groups. And we've collected a group of 40 healthy controls as well, who are age and gender matched to these individuals. So they're having these multimodal measures of inflammation, brain scanning, CSF and blood, and we're looking to see whether we can pick up an inflammatory signal which precedes dementia in this high risk group. So this is a work in progress, but I'm just sharing with you some of the baseline data, which we're just preparing, or we just submitted actually for publication. So we're looking here at the PK11195 signal, so the binding potential. The figure here shows that the mean PK signal for each of the groups, the controls, the low risk of dementia and the high risk of dementia, overlaid onto a structural brain template. And the color indicates the neuroinflammatory signal. So if we just look through and focus on a particular brain region, so just looking here at the putamen, you can see that it's blue, which means a very low inflammatory signal. And as we move through into the low risk and then the higher risk patients, it sort of warms up. So it's moving from blue through to green, indicating we can see more putaminal inflammation in these high risk cases. The same is true of frontal cortex. If we look at here, starting off green in the controls and warming up as we go through to the high risk cases. And the same is true if we look at hippocampal regions there as well. So it looks like there is more inflammation in those high dementia risk cases in multiple brain regions before they develop the dementia. So suggesting, as I said, that inflammation is preceding the emergence of cognitive dysfunction in these individuals. Now, in addition to that link between regional brain inflammation and dementia risk at a group level, we've also found correlation between cognitive performance and inflammation in multiple regions of interest. So for this, I'm showing you correlations between scores on the Adam-Brooks cognitive examination, which is a measure of global cognition, and PK11195 binding potentials across multiple brain regions. So these are all the brain regions where we see a significant correlation. And noticeably at the top there is the amygdala, so the same region that we saw in the post-mortem studies that seem to be, in that region there seemed to be a link between inflammation and dementia. Again, we're seeing that this is the region in which there's the strongest correlation with between inflammation and cognitive performance. And I should just remember to acknowledge at this point Nina and Lenny, who have collected the data and done all the analysis for this study. So I mentioned that in these individuals who are having brain scanning, we're also collecting blood to look at inflammatory markers. So we've started exploring whether there are links between the peripheral inflammation and what we're seeing in the brain. And that does seem to be the case based on the data we've looked at so far. So in the serum, in the blood, we've measured a panel of 5 different cytokines. And for 3 of those cytokines, there are significant correlations between their levels and the total brain, average brain PK signal. So that's the case for IL-1 beta, for interferon gamma, and also for IL-6. Okay, so I've explained that there's evidence that there's more inflammation in the blood and in the brain in Parkinson's disease. And that seems to be linked to progression and to dementia risk. And we've also seen that peripheral immune cells can infiltrate into the brain. So the next question is, which immune cell types are actually involved in this and which are most relevant in terms of PD pathogenesis? So we're exploring this with some detailed immunophenotyping studies in our high and low risk, high and low dementia risk groups. And I'll take you through some of that data now. So we've been doing immunophenotyping using flow cytometry to look at multiple different immune cell subsets. And I'll start by just taking you through the data on monocytes, which are the key innate immune cells that provide the first response to infectious pathogens, and are also aggravated by aggregated proteins, including aggregated alpha-synuclein. Monocytes are typically separated into these three groups: classical, intermediate, and non-classical, based on their expression of 2 surface markers, CD14 and CD16. And the classical monocytes are thought to be the most inflammatory. They are most phagocytic, and they produce they're the sort of biggest producers of pro-inflammatory cytokines. And what we found in a cohort of 40 PD patients versus 40 controls was that classical monocyte numbers were increased in PD, and that seems to be driven particularly by this high dementia risk group. And this has been a consistent finding, certainly the PD versus control finding in a number of studies from different labs. Now, I mentioned earlier toll-like receptors 2 and 4, and the fact that these receptors seem to be important in terms of mediating the innate immune response to alpha-synuclein. And so we looked at these expression of these receptors on the monocytes from the patients as well. And what we see is that there seems to be an increased number of cells expressing TLR2 and an increased number expressing TLR4. And again, just the same as with the shift towards the classical monocytes, in terms of this increase in TLR expression, it seems to be mainly driven by those individuals who have a higher risk of dementia. So this, the other thing to say about TLR4 is that it is otherwise known as the LPS receptor. So we know that it also mediates the response of these cells to endotoxin. And what we find in the Parkinson's disease blood is that there seem to be elevated levels of serum endotoxin. So another theory, in addition to the idea that immune cells are activated by alpha-synuclein, is that they may be activated by elevated levels of endotoxin coming from the gut. There is a whole story, as I'm sure many of you will know, around the gut in Parkinson's disease, and the idea that gut inflammation may be a really early feature of the condition. It's quite an involved story, which I don't have time to go into today, but that's just interesting to just flag up this point that endotoxin levels do seem to be higher in the Parkinson's cases. So Another cell type we've been interested in recently are natural killer cells. These are part of the lymphocyte family, but they are also part of the innate rather than the adaptive immune system, unlike other lymphocytes. And a key role of these cells is to target and destroy either damaged or malignant cells. They're a rather understudied cell type in Parkinson's disease, and we've only just started looking at them, so the numbers here are rather small. We've just got data on 10 PD cases and 8 controls. And with this flow data again, we gate these cells according to their lack of expression of CD3, which is a T cell marker, and their expression of CD56, which is the classic NK cell marker. So in the PD cases, there are more of these NK cells. And then this is work done by Johnny Holbrook, a postdoc in my lab. He found that in addition to that, there are more of these NK cells in PD. There are more of a particular subset of them that are this mature cytotoxic subset, which express CD16. And also there seems to be increased expression of a marker NKG2D, which is known to be an activation marker on NK cells. So as I say, the numbers here are rather small, but suggesting that there seems to be a shift towards more activated NK cells in Parkinson's. What about the T lymphocyte component? Well, our main finding in terms of T cells in Parkinson's has been that there seems to be a shift in senescent cell subsets. So I'm going to do a brief sidestep at this point, just to talk very briefly about aging and the immune system. Which is potentially highly relevant given that Parkinson's is a disease of older age. So in the normal in normal immune aging, we know that there's involution of the thymus, and we think that this, in association with repeated exposure to viral infections throughout life, contributes to a shift in the profile of T cells, particularly CD8 or cytotoxic T cells. So what we see with advancing age is a drop in the number of naive T cells and an increase in the number of these differentiated effector memory cells. So these cells are less good at responding to novel antigens, but they can produce a highly cytotoxic response to a restricted pool of antigens that they recognize. And this is what's thought, this shift from naive to these highly differentiated effector memory cells is what's thought to underlie the reduced response to infections, to new infections, and the reduced response to vaccinations in older people. And also to underlie the increase in autoimmune responses that we see in older people. So we thought it might be interesting to look at how whether this shift was altered in Parkinson's disease, given that, as I say, it is a disease which is more common with advancing age. And we were surprised to find actually that we thought what we might find was that we would see more of these differentiated effector memory cells, that we would see sort of accelerated immune aging. Actually, we seem to see the opposite. So a drop in these senescent CD8 cells. And we've characterized them in 2 different ways using different cell surface markers, of which the details don't matter too much, but the basic message is that there's a reduction in these senescent cells in PD. And we've replicated that now in 2 separate cohorts. And we don't really understand quite what this means, but we hypothesized that it might underlie the more active response to novel antigens, for example, aberrant alpha-synuclein in PD. We've also found, or rather Nina has found, as she's been doing this work, that there seems to be an increase in the infiltration of CD8 T cells into the CSF in Parkinson's disease, and it's possible that that might underlie what we're seeing in the blood with that drop in highly differentiated senescent cells. It might be that they're transiting into the cerebrospinal fluid. Because the numbers of T cells in the CSF are quite small, it's difficult to be very certain about the exact subtype of those cells, but we do see more CD8 T cells transiting into the CSF in PD, and more of them express some of these markers, senescent markers, such as CD57. So that's a hypothesis that we're exploring further, the fact that senescent cells, or rather highly cytotoxic cells, might be trafficking into the CNS. What about CD4 T cells? Well, the main finding in terms of CD4 cell subsets is that we see a drop in the percentage of regulatory T cells. So we looked at regulatory T cells using various different surface markers, but and actually intracellular markers. but the message seems to be that the same, that we see a drop in Tregs in Parkinson's disease. We also see a drop in Th2 type CD4 cells, which are the more anti-inflammatory cell subset, as opposed to Th1 and Th17, which are more pro-inflammatory. And this shift in terms of a drop in Tregs, again, it is a finding that's been replicated by others in the literature. So we think this is a real thing, and suggests a shift towards a more activated CD4 response in PD. And then final cell type I'm going to talk about the B cells, and this is going to be a busy slide, but it's a summary of a large body of work from Kirsten Scott, who finished her PhD in my lab a few years ago and is now a clinical research associate. So Kirsten looked at B lymphocyte numbers, first of all, in Parkinson's disease, and found that they were reduced. So just to orientate you here with the colours, the blue are the controls and the red are the Parkinson's cases. This is, again, in that same cohort of 40 patients, 40 controls. So we can see here a clear drop in terms of overall number of B cells in Parkinson's. And here, these grey and black subsets are the PD patients that are stratified according to their dementia risk again. So here we have the low dementia risk, an intermediate dementia risk group, and a high dementia risk group. So we can see that, again, this effect is particularly driven by those individuals who have this more malignant phenotype and are more likely to go on and develop a dementia. Of the B cell subset that B cell subsets that Kirsten has explored, the subset that was specifically reduced in number were those expressing a marker called CD1D, and these are cells which are thought to have a regulatory function. She also found that lower regulatory type B cells were linked to worse motor scores, so more motor impairment, suggesting that these B regulatory cells might have a protective role in Parkinson's. So they might be important in terms of dampening the immune response and reducing neuroinflammation. And in keeping with the idea that some B cells might be protective, she found that depletion of B cells in an animal model seems to exacerbate pathology. And then the other sort of B cell related marker that she's been looking at are antibodies. So we know B cells have an important role in terms of antibody production. So she looked specifically at antibodies to alpha-synuclein. And she's been doing that not only in PD cases, which are in red here, but also in individuals with a condition called REM sleep behavior disorder, who we know are high risk of going on to get Parkinson's disease. So what she found was that in a group of individuals who have this condition called REM sleep behavior disorder, along with other markers that suggest that they're particularly close to converting to Parkinson's, there seems to be a peak in alpha-synuclein antibodies. So Kirsten's data backs up this idea that the immune response in PD might be partly driven by alpha-synuclein, and to supplement that, she then did a meta-analysis of the literature, which confirmed this idea. But it seems that this elevation in alpha-synuclein specific antibodies is only there in early stage disease. So this is data looking at Parkinson's cohorts that are less than 6 years from diagnosis. And there we see this association. But if we then look out at individuals with later stage disease, the association with PD disappears. In terms of T cells, David Saltzer's lab in the States have reported a finding of alpha-synuclein specific cells, which occur at significantly higher numbers in Parkinson's cases versus controls. And again, this seems to be a feature of just early stage disease, which seems to wane later on. So here, if you can see this data, if you have a big enough screen, you'll be able to see that along the x-axis here is years from diagnosis. And this is a production of inflammatory cytokines in T cells that are stimulated with alpha-synuclein epitopes. And we can see here that in early stage disease, so up to 10 years from diagnosis, you seem to get this cytokine response. But in people who've had Parkinson's for much longer, that response is not there. They replicated the same thing here in a second cohort, and this is the combined cohort. So quite clearly showing that this, at least the T cell specific response to alpha-synuclein is there in early disease. And this is really important for us in terms of planning clinical trials, because it would suggest that if we're going to intervene with anti-inflammatory or immune modulating therapies, we need to be doing that early on. So down to my final question, how do we translate these findings into clinical trials? So going back to this hypothesis that peripheral immune activation might be driving central pathology in the brain and leading to disease progression, I've discussed with you evidence to support this idea. So we talked about the fact that not only brain inflammation, but also peripheral immune activation are there and are associated with a more malignant phenotype and with more rapid progression. And we talked about the fact that in terms of the drivers of that immune response, alpha-synuclein is implicated, endotoxin is also implicated, and this might be linked to gut inflammation and early gut pathology in Parkinson's disease. Now, one of the things you might have spotted as I talk through the work on peripheral immune activation is that there are a large number of changes in different arms of the immune system, and multiple cell types are involved. And we've still got quite a lot of work to do in terms of untangling which components of the immune response are the most critical in terms of mediating the pathogenesis of PD. So in terms of moving to clinical trials, we decided that perhaps a good idea in the first instance would be to use a rather broadly acting immunosuppressive drug to knock down multiple peripheral immune cells and test the proof of concept that immune activation is playing a role in driving all of this in disease progression. So we have started a trial called AZA-PD using this drug azathioprine, which is a peripherally acting immunosuppressant. It is already widely used in a number of inflammatory conditions and things like inflammatory bowel disease, rheumatoid arthritis. We know it inhibits nucleic acid synthesis and it reduces leukocyte proliferation. So it will reduce a number of different immune cell types. And the objectives of this trial are, first of all, proof of concept. So we want to see whether if we use a rather broadly acting immunosuppressant drug, knock down all of that peripheral immune activation that we're seeing, will it impact on clinical progression over time? And secondly, we want to look at proof of mechanism. So can we demonstrate that if we knock down peripheral immune activation, we will have an impact on immune cells infiltrating into the CNS, and we will be able to have an impact on brain inflammation. So this is how the trial looks. So we're recruiting people with early stage Parkinson's disease. I mentioned earlier that we seem to see this peak in the immune response in early disease, which goes away later on. So it's important that we go for early stage cases. They have no concurrent immune conditions. They're randomized 1-to-1 to receive either azathioprine or placebo for 12 months. We are collecting blood and CSF both pre and post treatment to look at changes in serum immune markers, but also changes in terms of infiltration of immune cells into the cerebrospinal fluid. And we're using PET imaging with this PK11195 ligand to look at brain inflammation pre and post treatment. And the patients are then followed up 6 months later to see whether any effect is consistent after removal of the treatment. And our primary outcome for this trial is a clinical one, so we're looking at change in a movement score. So we started the trial back in April of 2021, and I'm pleased to say that we managed to recruit the full cohort. In fact, we slightly over-recruited, so we recruited 67 participants to the trial who are now either have completed or are still receiving their treatment. And we will have some answers, hopefully in about a year's time, as to whether we can actually make an impact on progression of Parkinson's disease by suppressing the immune system. Now, in the meantime, there are multiple other immune-based therapeutic targets that are under consideration. So phase 1b trials have recently been started of an inflammasome inhibitor, and there are other inflammasome inhibitor trials planned, which will well, the aim is that they will knock down the innate immune response in terms of reducing microglial activation and also reducing peripheral monocyte activation. There is considerable interest in using drugs which can block toll-like receptors. So there's particular interest in this drug candesartan, which is an antihypertensive therapy, which is known in vitro to be able to reduce expression of TLRs. So this is a very attractive potential drug to use for Parkinson's, because we know that it's safe to use in an older population. So it could be rapidly repositioned for a PD trial. There's interest in manipulating the adaptive immune response. I talked about the fact that T regulatory cells are reduced in number. They also seem to be reduced in function in Parkinson's disease. And a phase 1b trial has already been done a couple of years ago of an agent called salgaramestin. And they have shown that this can boost the number of T regulatory cells and boost their ability to suppress the T effector cell response. And there was a suggestion that there may also be a clinical improvement with this drug, but the numbers were rather too low to draw any definite conclusions on that. Other strategies which we might think about will be using something like low-dose IL-2, which we know can also boost the number of T regulatory cells. We may be able to target molecules which are involved in immune cell trafficking. So reducing trafficking of monocytes and T cells into the CNS is a very attractive therapeutic strategy. And then I briefly touch on this idea that the gut and gut inflammation might be important. And there is considerable interest in manipulating the microbiome using either probiotics or even using fecal transplants. And then another potential target would be blocking the endotoxin itself that's produced by the gut bacteria. And in animals, LPS vaccination is a thing, so you can vaccinate cows against LPS, and that's something that people have sort of vaguely considered as an option in Parkinson's disease. So that may be a future consideration. Okay, so I'll summarize just by saying that genetically determined immune variation and the use of immunosuppressant drugs seem to be able to alter your risk of developing Parkinson's disease. We know that in established Parkinson's disease, there is immune activation not only in the brain, but also in the periphery. And immune activation seems to be linked to clinical phenotype and also to a more rapid progression of disease, suggesting it's not just a bystander effect of a neurodegenerative process, but it might have something to do with driving disease course. We know that peripheral blood inflammatory markers seem to correlate with brain inflammation. We know that the immune response in Parkinson's disease includes changes that are quite complicated and involve both innate and adaptive immune cells. And we know that both peripheral, innate, and adaptive cells can infiltrate into the central nervous system. So it's a rather complicated and messy picture, but the AZA-PD trial has been started to just test this concept that overactivation of the peripheral immune system is playing a role in PD. And we will hopefully know, as I say, in a year or so, whether azathioprine can have any impact on slowing PD progression. But there are multiple other more targeted approaches that will no doubt come into clinical trials in the next few years. And I just want to finish by thanking members of my group who are pictured there, all of my research colleagues, all our research participants, without whom we wouldn't be able to do any of this work, and all of our funders. So thank you. Thank you for listening.
Thank you, Caroline, for a wonderful talk and a fantastic finish to an excellent term of seminars. Thank you to all our speakers this term, and make sure you catch up with all that you've missed on our YouTube channel. For more info on the talks covered in this seminar series and all things neuro-related in Cambridge, please follow us on Twitter @CamNeuro and follow the links below. See you next term.